Welcome to the siege of Myocardia Castle. With the help of invading soldiers and a herd of highly prized cattle, we will cover everything you need to know about excitation-contraction coupling in cardiac muscle, the process by which an action potential transmitted to a muscle cell is translated into the production of tension. But before we can get into that process, you will first need to know a little bit about the myocardial cell structures that make it possible. This includes the sarcomere, the contractile unit of the myocardial cell, which is made up of thick filaments of myosin and thin filaments of actin, tropomyosin, and troponin. These filaments overlap in a way that is reminiscent of the thick and thin planks making up this drawbridge. Actin and myosin have complementary binding sites that allow for the formation of specialized actin-myosin crossbridges. These are represented by our now opening drawbridge, as it will soon serve as a crossbridge, because you will be able to cross the bridge. Get it? When these actin-myosin crossbridges form and then break, the thick and thin filaments will slide past each other, causing the muscle fiber to shorten and generate tension. This cross-bridge cycling will typically be inhibited by tropomyosin, a coiled protein intertwined with actin that blocks the myosin binding sites. This is represented by this filament-like chain, which is used to hold the drawbridge up. In essence, it inhibits the drawbridge from opening, which prevents it from becoming a cross-bridge. Troponin is able to remove this inhibition by binding with calcium and undergoing a conformational change that moves tropomyosin away from the myosin binding sites. This, in turn, allows for actin-myosin crossbridge cycling to begin. Kind of like how this tropical troll bound to a cow can break this chain and turn a drawbridge into a crossbridge. Next up are intercalated discs, which maintain cell-to-cell adhesion between myocardial cells and contain gap junctions. Gap junctions are vital because they are low-resistance pathways through which action potentials can rapidly spread from one cell to another. Not unlike how this gap in the wall is offering a low-resistance pathway to this action hero-like soldier. Gap junctions are what enable the heart to behave as an electrical syncytium, which is a fancy way of saying it can function as a unit. Next are the T-tubules, which are invaginations of the myocardial cell membrane that allow action potentials to be carried into the cell's interior. Hence these invaders rushing in through a T-shaped doorway. They also interact with the sarcoplasmic reticulum by forming a dyad. More on this in a little bit. Speaking of the sarcoplasmic reticulum, it is a series of tubules inside the cell that store and release the calcium necessary for excitation-contraction coupling to occur. This pen, being used to store cows inside the castle yard, should help you remember this. These tubules are located right next to the sarcomeres, the cell's contractile elements, which puts them in close proximity to the calcium binding sites on troponin. With that out of the way, let's break down the steps involved in translating an action potential into a contraction. Once an action potential has been initiated in the cell membrane, the first step is for the depolarization to spread to the T-tubules, where it will be carried into the cell interior. During the plateau phase of the action potential, calcium conductance through voltage-dependent L-type calcium channels, also known as dihydropyridine receptors, will increase, creating an inward calcium current. This invader has opened a gate and let a cow charge into the castle yard to help you remember this. This slight increase in intracellular calcium, in turn, triggers the release of even more from the sarcoplasmic reticulum via calcium release channels, also known as rhyodine receptors. This process is unimaginatively known as calcium-induced calcium release. You can see this depicted by our inward-running cow that has now broken open the pen, inducing the release of even more cows. The amount of calcium released during this process depends on two factors: the amount stored in the sarcoplasmic reticulum, and the size of the inward calcium current that triggers the release. This release leads to a significant increase in the intracellular concentration of calcium, or cows on the loose, which increases its binding to the troponin C subunit of the troponin complex. Because of this, troponin is more likely to undergo the conformation change that will remove tropomyosin's inhibition of actin-myosin binding. This allows actin and myosin cross-bridging to occur. Remember our Castle's cross bridge? The thick and thin filaments to slide past each other and the myocardial cell to contract. This cross-bridge cycling, which is fueled by ATP, will continue as long as the concentration of calcium is high enough to keep the binding sites on troponin C occupied. Thus, the magnitude of tension generated by these cells is proportional to the intracellular calcium concentration. This also means that the relaxation of the cell will occur when the intracellular calcium concentration decreases enough that it will dissociate from troponin C, freeing tropomyosin to once again inhibit cross-bridge cycling. This decrease is facilitated by two different transporters in the cell. The first of these is the calcium ATPase pump, or SERCA, which reaccumulates calcium in the sarcoplasmic reticulum. Kind of like how this guy is trying to reaccumulate the cows in another pen. The second is the calcium sodium exchanger, or NCX, which moves calcium out of the cell in exchange for 3 sodium ions. In its haste to escape, this cow has knocked a barrel of salty peanuts into the castle yard to help you remember this. And that's it. Before we wrap up and escape, let's take one last look at what we've learned. Excitation-contraction coupling is the process by which an action potential in a myocardial cell is translated into the production of tension. When an action potential is initiated, the depolarization spreads from the cell membrane to the cell's interior via T-tubules. This, in turn, increases calcium conductance through voltage-dependent L-type calcium channels, aka dihydropyridine receptors, creating an inward calcium current. This then triggers calcium-induced calcium release from the sarcoplasmic reticulum, greatly increasing the intracellular calcium concentration. Troponin C can then bind to this calcium and undergo a conformational change that will remove tropomyosin's inhibition of cross-bridge formation. With tropomyosin out of the way, actin-myosin cross-bridge cycling in the sarcomere can occur, and the myocardial cell will contract. For the cell to relax, the intracellular calcium concentration needs to decrease enough for calcium to dissociate from troponin C. This is facilitated by the calcium ATPase pump, SERCA, which reaccumulates calcium inside the sarcoplasmic reticulum, and the calcium-sodium exchanger, NCX, which moves calcium out of the cell in exchange for 3 sodium ions. Now, if we want to avoid getting trampled by these cows, we better get a move on. Yeah, I'm sorry about that one. We'll see you next time.